Hey ANPers, this is Dr. Jones checking in. This is ANP2, lecture test 6, video, checks notes. Last one was 9, so this one must be 10. I'll write that down. I gotta keep track or I won't remember which pages we did in which video. So last time we went from 15 to 18. I must have gone over time. So we are starting in the middle of page 18. Here, early embryogenesis, gastrulation, and germ layer formation. All right, so gastrulation, first you form the yolk sac. That's right, you had a yolk sac, just like a chicken has a yolk sac. The birds actually develop in the white, not in the yolk. They eat the yolk just like you ate yours. Quail eggs too. Quail eggs are way tinier than chicken eggs. They're like, it takes like 3 quail eggs to make a chicken egg. Pickled quail eggs are surprisingly tasty. They're like just a little, like a protein snack. I am super fond of the pickled quail eggs. Mostly we're keeping the quail for meat, although we haven't processed any yet. And so the extra eggs we have while we're waiting for the eggs in the incubator, we're pickling them, and they're really good. I'm surprised. Way better than I like pickled chicken eggs. Pickled chicken eggs are just so huge and like a lot of yolk. And quail eggs have more protein per like amount than chicken eggs. Quail meat has more protein than chicken, which is weird. Okay. All right, we were making videos about anatomy and physiology. We should get to that. So I am at the term gastrulation, which is this term here. We've talked about the yolk sac. So it was related-ish. Okay, so gustation is, gestation is, preggers, and gastrulation is before you were born. Okay. So all right, back up. Zygote, single cell, cleavage, and then you were 2 blastomeres, 4 blastomeres, and then you were a morula. And then you became a hollow ball of cells. The hollow ball of cells entirely is the blastocyst. The hollow part is the blastocele. The cells on the outer edge are the trophoblast. These are all lab terms you must know, and for lecture 2. And then the inside, the light blue bits, are the inner mast cells. That was you. You were that little clump of cells on the inside of a hollow ball of cells. That's where you come from. All of us. All living animals, actually. And so then that implants, you implanted into your mama's uterus. Tell her thank you. And then the inner mast cells become 2 layers of cells, and we're learning those terms for lab. And then you've got the cellular trophoblast and then the syncytial trophoblast, which again, lab terms. So then these 2 layers, those are not the terms. We're learning hypoblast and epiblast. Then they become the ectoderm and the endoderm. And this is a cross section. If you're looking down on it, it makes an oval. And so it's called the embryonic disc. So the ectoderm down the middle, here's the oval, down the middle of the oval folds in. Kind of like a coffee bean. Folds in, except not round. Yeah, not like a coffee bean. Folds in and forms a 3rd middle layer of cells. And the middle layer of cells is the mesoderm. Meso, like Mesozoic era, or like a metaphase plate. Meso. We had meso somewhere else. We've had meso before. Not miso, that's soup. Also good. Meso means middle, and forming this middle layer of mesoderm is gastrulation. So gastrulation is a process, like implantation is a process, not a structure. So gastrulation is forming the mesoderm, which is the middle layer. Okay. So the three layers together are the germ layers, the ectoderm, mesoderm, and endoderm. And those are the primary germ layers. And the three germ layers are the baby, which is the wrong term. We're implanted, so we're at embryonic development. So this is an embryo because we have implanted. And then the embryonic disc is this whole oval. It's cut in half here. The embryonic disc is the oval with the three germ layers. The germ layers are these, because these cells divide so much, they're germinative cells. Like the stratum germinativum is the other word for the stratum basale, the basal cells, the stem cells in the skin. And so these are considered germ layers because they create so many more cells. Okay, and so here's the whole thing together from the embryo plates. And then here you can see the intermass cells, and then the, it doesn't show the gastrulation, but it does show the three germ layers. And you can see here the ectoderm becomes the neural tissue, mesoderm becomes connective tissue and muscle tissue, and then all three layers make the epithelia and glands. So these are where all of your tissues come from, embryologically speaking. Ooh, that's a fun picture. Little baby. Okay, so then here you are at week 3, that disc, the front of it kind of folds under, and that is the head fold. Do I have a bigger picture of the head fold? No. I need a better picture of the head fold. That's teensy. Okay, so week 3-ish we have the head fold and it makes the head. And then at week 4-ish you have the tail fold, head fold, embryo. Size and shape of a cashew, then it's an embryo. Again, implanted, it makes the embryo. So tail fold week 4, and that makes not your legs but your bum. Your bum is your tail. And then, yeah, okay. And then those keep developing, and then let's talk about the extra embryonic membrane. This is, I had a slide for this. I think this is the wrong PowerPoint, the wrong version of the PowerPoint. Okay, so the extra embryonic membranes are everything in here that's not the embryonic disc. So everything in here that's not the embryonic disc. So the yolk sac, the amnion, which we're familiar with, the chorion, and we will say, whether it's this video or next, at some point the chorion will grow out to the amnion. Let's see, here's the amnion. Oh, they're already fused here. The picture is so tiny. Okay, so here's the amnion and here's the chorion. And when they fuse, they become the amniochorionic membrane. It's science. You take the two words and you stick an O between them and you call it a new word. I-O-N. That's an R. Amniochorionic membrane. And the other is the one that's, it looks French, so how do you pronounce it? Elantois. Anyway, that one becomes a urinary bladder. So that's handy to have around. The chorion, this one, has an early role in the maternal embryo exchange of nutrients, gases, and wastes. The chorion on the side towards the implantation will become, isn't yet at this point, but will become the placenta. And so before the placenta is developed, it's the chorion that does the exchange of nutrients, gases, and wastes with the maternal blood supply. Page 19. Placentation, oh, here are the terms: yolk sac, amnion, allantois, and chorion. I didn't have to write them all down. Okay, so placentation is when you grow a placenta, when that chorion, the chorionic villi, become a functioning placenta. The embryonic heart, which is right there, starts beating at the 3rd week of development, which is the 5th week of pregnancy. But it's not a heart, and it's not beating. So they are heart cells. It's not quite pumping blood through vessels to all the cells yet, but they are heart cells, and they are sparking. And there's not chambers where it's like a coordinated pumping of blood. It's not a functioning heart like an actual born child, but they are cells that have heart function, and they are sparking. So that happens. The blood in the umbilical arteries is deoxygenated. That's the next picture. There we go. The umbilical arteries is deoxygenated and has wastes. And there's a reason for this, because before you were born, you did not get oxygen from your lungs. You did not get oxygen from your mouth and nose. You got your oxygen from your belly button. I'm pointing at my belly button. This camera doesn't show low enough. Because that's where the umbilical cord is. It goes in your belly button. That's what the belly button is, the umbilicus. And so you got your oxygen from the placenta. And so when you were big enough to have a placenta and a heart that's beating through arteries, then the blood going out from your body to the placenta and arteries go away from your heart is carrying deoxygenated blood to the placenta, because the placenta is where you get oxygen from mama's bloodstream. You just suck it right out. As you may recall, fetal hemoglobin has a much higher affinity for oxygen than adult hemoglobin. So even though it's mama's fully oxygenated blood, you just suck oxygen straight out of it. Yeah. So that's why mamas are so tired. Because babies are drinking all their oxygen. And so then the umbilical vein, there's 2 umbilical arteries and 1 umbilical vein. The umbilical vein is red oxygenated blood. So arteries are red and veins are blue, except, and we have 2 exceptions now, the pulmonary system and the umbilical arteries and veins, the umbilical vessels. There we go, that's the term. All right, so fetal hemoglobin strips oxygen from the maternal hemoglobin because it has a much higher affinity for oxygen, which I said about a minute too soon. And what separates maternal and fetal blood? Layers of the trophoblast. So mama's blood and baby's blood do not mix unless there is a tear and bleeding at the placenta, which can happen in things like placenta previa. But normally, mama's blood cells stay in mama's bloodstream and baby's blood cells stay in baby's bloodstream. What crosses are the gases, the nutrients, the waste, some antibodies, and lots of toxins. So it's during pregnancy that many women find that they want to be more cautious about what chemicals they interact with. I mean, everything's a chemical. Water's a chemical. You want to interact with water to a degree. Don't drown it. but there are things that cross the placenta that aren't good for baby, especially the smaller baby is, the less ability they have to filter out things that are harmful to them, things that wouldn't hurt you in the same degree. So, yeah, it's a thing. Okay, so organogenesis, I think is the next slide. There we go. Look, there's a little disc and the little head fold and the little tail fold. And then look, the pharyngeal arches. And now if you were a fish, these would become gills, but you are not a fish. So this becomes your maxilla and your mandible and your hyoid. So this is your lower face right here. And there's your little eyeball and your ears way back there. And your eyes are like way off the side of your head and they have to like rotate in. It's all about differentiation and movement. The cell clusters move to where they need to be. So there's the two parts of the heart, and then the somites. The somites will become your backbone, your vertebrae, and also the muscles. So, and there's a little arm bud, and the arm bud grows before the leg bud, and the tail is your bum. And so, and then here, week 8, so this is still an embryo at week 8. It's almost looks human, but it's not yet a fetus. So, and you can see the ambion, the umbilical cord, and there's 12, and that is weeks 9 through birth are, of course, fetal development, and week 4, again, size and shape of a cashew, so that's an embryo. Here's still an embryo. Look at the little arms and the little legs. And the eyes are starting to rotate in. Ears are still way out here. Does it show the nostrils? Nostrils start way off to the side too. Okay. Did I put a timer on? I did. We have 2 minutes left. Okay. We will finish this page then. Organogenesis. I swear I have a version of this note set where all these words are typed in. I miss it. Organogenesis is organ formation, and by the end of the first trimester, you have the beginnings of all of the major organ systems. So here's another picture of the disc with the head fold and the tail fold. And then here, oh look, there's the ear. That's that same picture, but a drawing. The pharyngeal arches. And there's the whole gut from mouth. The intestines, the small intestines at this point are actually in the umbilical cord, so that's why it goes out. Okay, and there's a 4-month fetus, and then 6-month, this was, I think, the 2012, so this was before they had the 3D. I think now they've got a 3D picture. Okay, over the second trimester, the fetus grows to 1.32 pounds, which is very specific for an average. When the amnion contacts the chorion, they fuse and become the amniochorionic membrane, which we've talked about. During the third trimester, the organ systems get ready to function. So let's finish this page, and then we'll call it a video. We did, so that'll be 18 and 19. On this video. Okay, I got that written down, because I will not remember. Okay, so the human chorionic gonadotropin, which I have written down for you, and I don't see on this picture. Why is it not on this picture? Okay. Is in the blood. It is positive for pregnancy. And it acts like luteinizing hormone after implantation. Okay. Human placental lactogen is from the placenta, and it's a lactogen, so it makes milk. Ogen is for make, and lactose is milk sugar. Placental prolactin is placental, so it's from the placenta, and it's a prolactin, so it also makes milk. The relaxin increases the flexibility of, there's our timer, increases the flexibility of the pelvis and in fact all of the joints. So that's part of why women who are big pregnant kind of waddle, because all of the tendons and ligaments that knit those joints together have been relaxed by the relaxin. So there's more play in every joint, which makes room for baby to get out, but also makes us a little unstable because all of our joints have a little extra range of motion. And progesterone is a hormone that maintains the pregnancy, and estrogens are the hormones that help stimulate labor and delivery. You may recall the main estrogen is estradiol. All right, so we are going to call that, I had a post-it there, back on page 18. So we're going to start on the top of page 20 for our next video. And I think I've lost the light anyway. It's no longer daylight. It's dusk. Like the sun is set, but there's still some late clouds. They're kind of orange and a little pink. So sunset is always fun. So this has been ANP 2 lecture test 6 video. I've forgotten already. This one was 10. 10, oh my goodness. And we covered pages 18 and 19, and we will start with 20 for the next video. So thank you as always for tuning in. The education must go on. Dr. J out.